this brief recording really is an attempt to give a very brief overview on SARS-CoV-2 based on the paper that I'm currently writing. And my paper really focuses on the molecular pathogenesis, the inflammation, particularly non-specific, and indeed the immune responses following infection with SARS-CoV-2. And SARS-CoV-2 obviously is a virus from the coronaviruses that causes a severe acute respiratory syndrome, hence its name, SARS-CoV-2, severe acute respiratory syndrome, coronavirus 2, because not so long ago we had coronavirus 1, which caused the infection that we know as SARS, the original SARS infection. So SARS-CoV-2 itself causes a disease called COVID-19. And really, with the paper that I'm writing, because this is just a very, very brief overview, my interests really have been based on, how the virus infects us as the host and why it's so efficient at infecting us as the host. And the reason for that really is that SARS-CoV-2 and the original SARS both target an enzyme receptor that we express as human beings that is called ACE2 receptor. It's essentially an angiotensin enzyme converting receptor, which genetically it's found on the X chromosome, specifically the short arm of X chromosome. Is it Q or is it the long? I can't remember. Well, it's found on the X chromosome. Let's see, does Q22 look better? It's found on the X chromosome anyhow. Either one of the arms of the X chromosome. And obviously we all have X chromosomes, therefore unless we have mutations in the region of the X chromosome, then we will be able to express ACE2. So ACE2 is an enzyme actually. So it's an enzyme that converts angiotensin into an ANG1 to 7. And again, I'll look at that briefly, because I have looked at it anyhow. So these receptors are normally expressed on a number of cell surfaces, and they probably look something similar to this, really. And this is just called a claw-like protease. That's what a lot of people are calling it, because it looks like a claw in terms of that it has, sort of like a hook, really. And there are some specific amino acids, which I've included as part of my paper, really, that are recognized by SARS-CoV-2. And those specific amino acids cause us, to this virus to bind with a high affinity and specificity. And obviously, if it's binding with a high affinity, it means that it's binding incredibly successful, which is a new thing that hasn't been seen before. So it binds incredibly successful to the ACE2 receptor. And once it is bound to the ACE2 receptor, it then needs other enzymes of the human body, because although the virus, does encode its own, its own, so anti-immune mechanisms and defenses, it actually needs the human body in order to infect. And it infects the human body by using serine proteases, which are enzymes. Specifically, an enzyme called furin, and another enzyme which is called TMPRSS2, and that's just a transmembrane receptor serine protease 2. And both these two enzymes are required, and they will bind, transform, and activate the virus at different sites in order to facilitate its entry into the cell. So, for example, the virus itself is a coronavirus. It just has a single strand of RNA, it has a nucleocapsid, and it has an envelope and spikes on the outer attached to the envelope or membrane. So these spikes are called S proteins. The spikes. They're just made of a glycoprotein, really. And these spikes are what recognizes the ACE2 receptor. So the spike will recognize and again it recognizes some specific amino acids, and which cause it to bind quite efficiently. And once it is bound to the ACE2 receptor, if it essentially looks something like this, really. If we were to take one spike and open it up, so it's made of two subunits. The subunit which is called S1, really because it's an S protein, and another subunit which is called S2. So S1 and S2 have a junction here, and this junction has inserted, the virus has inserted an amino acid sequence, 4 amino acids sequence that are recognized by furin. So this is a furin recognition site. And the S protein also have what we call a fusion peptide. And this fusion peptide, obviously, as the name indicates, allows it to fuse with the host proteins. However, the important thing to know about this fusion peptide, indeed the entire S protein, is that it has what we call potential for activation. So it has activation potential. And what this means is that it can be activated And activation will then mean that it is ready to do something, because at an inactivated state, it cannot bind, it cannot enter the host. So it needs to be activated in order to enter the host. So activation results in host entry. And this activation is undertaken by these two enzymes that the virus desperately needs. And we express these two enzymes, we need them for a lot of systems in the human body. For example, furin itself is also used to activate one of the transformational growth factor, beta, which is really useful in activating, which is a powerful cytokine that can activate, immune responses. It's also really important in cancers, and studies have looked at whether it can actually be inhibited, whether furin can be inhibited, because obviously furin is actually has a role in the development of cancers. And in terms of inactivating furin, it's not so easy to inactivate an enzyme that is so essential to the human body. We express this enzyme all over our bodies. We require it for a number of processes. In terms of if we were to look at inactivating it, then it will mean creating synthetic inhibitors. There are some synthetic inhibitors that are out there, but at the moment they are not really suitable for oral administration. Also, furin itself, when it's expressed in the human body, it's expressed as a proenzyme attached to an autoinhibitor. And it would be interesting to check whether there is a mechanism to just inhibit it by using its autoinhibitor, but targeted at specific sites in order to block viruses that use furin. And indeed, it's not just SARS-CoV-2 that uses furin. A lot of processes, toxins, bacterial toxins use furin. Other viruses use furin. We use furin, of course, for a lot of our proteins that we need in the body. The other protease, there are inhibitors that have been found. For example, in Japan, post-operatively for esophagitis, you are given an inhibitor which is called Camostat, and it works really well. Also in Japan, they use it for chronic pancreatitis. So there already is an inhibitor for this that is utilized in treatments in other countries such as Japan. However, the only difference is that obviously, if you are only inhibiting one, the other is still active, and the human body does compensate for other proteins. For example, if you inhibit this protein, there are other certain proteins like cathepsin B and L, which will then compensate for this one. Therefore, even if you were to inhibit one without the other, you might not get the effect of inhibiting viral entry. So furin, just going back to where these enzymes actually work on the S protein, on the viral X protein. So furin will recognize this binding site there where this SARS-CoV-2 has inserted 4 amino acid sequences that are recognized by furin. So furin will come in, recognize the site, bind and remove S1. So this now means that this protein has been primed. Now it's primed and ready for activation. And then what happens is that as it's being primed, a conformational change occurs, allowing the other enzyme to come in and activate the fusion peptide. And activation, so this one will just come in and activate the fusion peptide. Activate fusion peptide. And what that means is that when you activate this fusion peptide, it then merges and fuses with the ACE receptor, so it becomes continuous. For example, say if S protein looks something like that as a spike, and we can see that the glial looks something like that. So these two just fuse into one membrane. And as they fuse into one membrane, obviously the virus is still here. And ACE, let me do it up here actually. So there we go. So we have our, oh, the virus is right. So we have the virus coming in. It's a small virus. It's very small in comparison to the human body. And we have our ACE receptor there. So the activation is occurred. So now we have a fusion peptide that has been activated. A conformational change occurs, causing the virus and ACE2 to fuse to have one single membrane. So because this is actually attached to a cell, so then we have that. This, the way I think about it is that it's sort of like going to eat it up and then bring it inside, like you are swallowing something. So it just eats up that virus because the two have fused and delivers it in. So the next stage will be, so ACE becomes like this. And of course we know that the virus will have spike. So we move from this to internalization. So once it has been internalized, the ACE protein becomes downregulated. So now in terms of just thinking about S protein downregulation, I think it's first of all is important to talk about where ACE2 itself is expressed. And ACE2 is a receptor that's expressed in a number of cells, but in my paper I'm actually mainly looking at the lungs. So in the lungs, there are studies that have found that the majority of ACE2 in the lungs is expressed in type 2 pneumocytes. And type 2 pneumocytes, their function in the human body, number one, is to produce lung surfactant, i.e. to reduce pulmonary tension and stop the alveolar collapsing against itself. And number two is to serve as progenitors for type 1 pneumocytes. And type 1 pneumocytes are the center for gaseous exchange. So now when we're talking about, you know, the alveolar, indeed we must just remember that. And there's a capillary down there. And this too becomes important in this. So now we know that this virus is incredibly clever in the sense that it will use ACE2 receptors that are found everywhere in the human body, including in the GI tract and the GU tract, actually in the brain as well, in the testes, in the kidneys. It's you know, all over the body really. It will use it to recognize specific amino acids that make it bind efficiently and makes it much more specific for this receptor. And once it has bound this receptor, it then uses these two enzymes that are found everywhere in the human body. We express them, we need them. Particularly this enzyme is expressed in type 2 pneumocytes. So type 2 pneumocytes are very well suited to being infected, because not only do they express ACE2, they have other serine proteases which are required by the virus, and they also have a lot of, you know, viral a lot of processes that are required for viral replication. So it will facilitate the mechanisms for viral replication. So type 2 pneumocyte is really well suited to being infected and targeted by ACE2. And the role of ACE2 in the human body, okay, we've covered this. So the role of ACE2 in the human body really is to control the renin-angiotensin system, whereby we have angiotensinogen, which is activated by enzyme renin into angiotensin 1, and then angiotensin 1 is converted by ACE into angiotensin 2, then angiotensin 2 is actually converted by ACE2 into Ang1 to 7, and indeed angiotensin 1 can also be made into Ang1 to 9, which eventually are synthesized into Ang1 to 7. So Ang1 to 7 itself is useful in the sense that it will bind the mitochondria assembly receptors and cause and reduce inflammation, reduce fibrosis, cause vasodilation, and really just have a protective effect. Whereas if ACE2 is left, there, nothing happens to it. If ACE2 signals, which it can do, it will bind, if angiotensin 2 signals, it will bind the angiotensin 1 receptors and cause inflammation. And when we say inflammation, it means non-specific inflammation that is quite destructive. And destructive usually because it's not specific towards anything. It wouldn't be specific towards the virus. It's non-specific. It goes everywhere. It targets everything that's in its way. It will also cause vasoconstriction, fibrosis, etc., etc. And of course, when we are having inflammation and we are of course I've already said it's non-specific and it's destructive, and it will also cause other things such as fluid to permeate, the interstitium. And if fluid is permeating the interstitium, eventually it will permeate the alveoli as well, thereby compromising gaseous exchange, which we need. So in terms of what the virus does, the virus will then downregulate ACE2. So this ACE2 coming in there, it means that it is being downregulated. So this is ACE2 downregulation. So when we're removing ACE2 in that short space of time as the virus is binding, it means that we're leaving angiotensin 2 to signal to work in the human body to bind its receptor to cause non-specific inflammation and all the other destruction. And once we have this destruction happening, and we must remember this is happening in the lungs, it means that we are not only causing destruction, we're also really, not only causing destruction, we're also compromising processes that should occur in the lung, such as if type 2 pneumocyte is being infected and being overtaken by the lung in order to do the lung's job of, propagating the virus so that it can disseminate, using our machinery to make more of itself and to stop all the processes of the human body, then it means that type 2 pneumocyte is not making what we needed to make lung surfactant. Also, really, normally when a virus infects a cell, the body itself causes what is called an antiviral state. So normally this cell, once infected, will be able to cause what is called an antiviral state. And in the lungs itself, ACE2 is considered to be protective in the sense that it will stop any injury happening due to this inflammation. Because say if there is a bit of inflammation happening, but there is ACE2, then ACE2 will quickly break down angiotensin 2, shunt angiotensin 2 products towards reducing inflammation, reducing fibrosis, and really being protective towards the lung. However, if you continuously downregulate ACE2, then it means that angiotensin is left free to cause a lot of inflammation. And as the virus is also downregulating ACE2, it then uses the human body machinery. Normally, when a virus infects a cell, the human body will express, will obviously use its own will then, express what we call interferon. stimulated genes, which then cause interferon to be expressed. And what interferon does, I can believe that I know. What interferon does is it works in blocking processes that the virus need to make more itself, and that blockage is essentially called inducing an antiviral state. So it will induce an antiviral state. And of course, the way this happens is you have a lot of factors such as regulatory factors, and then you also have transcription factors, which eventually then cause the transcription of interferons toll-like receptors, which will continuously block processes in the virus. For example, once this virus comes into the human body, it has to remove its membrane, remove the nucleus capsid in order to allow this RNA to be reverse transcriptized so that it can then enter the nucleus as DNA, or obviously, a reverse transcriptase enters the human cell, and then it can then be, more of it can be synthesized, so you can then amplify its synthesis, and then it will use our processes such as the ribosomes to then make more of itself virions, and then these little virions will eventually be packed into this process, because the virus will also use our ribosomes to make all of these proteins that it needs, the S protein, the membrane, the nucleocapsid, everything, and then its RNA will be packed again into little virions that then bud off. And as this virion is budding off, it obviously is taking a little bit of the cell with it and damaging the cell. So the cell will have holes and it will die. It no longer is useful for producing surfactant. So we've lost pneumocytes that can produce surfactant. And of course, it's important to know that it's not just one cell, one virus. A lot of cells, a lot of viral doors infecting, causing this, damaging the pneumocytes, going into our ribosomes, stopping the translation of all these proteins that we need, going into the nucleus, stopping the transcription factors such as nuclear factor kappa beta, such as interferon regulatory genes, IF3 and 7, from just entering the nucleus in order to create interferons that then cause an antiviral state. So it hijacks the system that will normally, induce an antiviral state, and then takes over so that it can then make more of itself, disseminate, infect more cells, the process restarts. So that's all really that happens. And one other thing, if ACE2 is removed and we lose this protective effect of ACE2, then angiotensin also will signal to stimulate